The final frontier may lie in the deepest reaches of the cosmos, but I'm here today to tell you that the next frontier is at the smallest of scales. Because we can see into the vastness of deep space and we can be awed by the motions of the heavenly bodies, but when it comes to observing the minuscule motions of life itself, we struggle to see the components involved. Because these components are smaller than the wavelength of light that we use to see them. And when these components are this small and you want to image a living cell on a microscope and your pixel size is just a single photon, well then anything smaller, such as DNA or proteins, become impossible to visualize. So how do we solve this problem? How do we monitor and measure the fundamental components of life at the scale of life? Because if we do this, I believe that we can unlock a new era in science and in medicine. And so my dissertation is focused on overcoming this very challenge, to be able to visualize the internal workings of a living cell. And my solution to this problem is to develop tiny nanoscale instruments that are capable of monitoring the local environment inside of a cell and then reporting that data back as a burst of light that is read by a microscope, very similar to the way a cell phone transmits its data as a burst of radio waves that's picked up by a tower. Now, one of the reasons that these nanoscale instruments can do what a microscope alone cannot is because they are on the scale with the size of the cellular components that they are used to observe. Just a few nanometers in size. To give you an idea of this scale, taking one of these nanoscale instruments and comparing it to the size of your cell phone, you will see that the same difference in size is equivalent to that cell phone and our moon. Billions of these devices can be working inside of a living cell without disrupting the ability for that cell to function. And so in my work, I deliver these nanoscale instruments into living cells where they work like a fleet of satellites to map out subcellular environments and to wirelessly report back information about their internal activities. And so when I use these devices, one of the examples that I've used them for is to monitor the delivery of therapeutically administered genes at the level of a single cell. And in the image I have behind me, one of those cells from those experiments is shown. And you may be able to make out the small green spots there. Now, those green spots contain these nanoscale instruments, and they are reporting on the location and status of the delivered gene as it makes its way through the machinery of a cell on the way to the nucleus in blue. Now, what this research demonstrates is that these nanoscale devices are capable of monitoring and reporting the location and status of a desired target. And these targets can be drugs, genes, or other proteins. But this is just a passive observation. And the next stage of this research is going to deliver more sophisticated devices that can be actively controlled to deliver a desired stimulus. So thank you for listening to my talk.